I'm gonna be real with you. I never understood the appeal behind having a sugar daddy. They're unreliable, they don't actually love you, and to them, you're replaceable. But there's one aspect of a sugar daddy that won't let you down, and that's the sugar. Although carbohydrates and sugars won't fill the lack of meaningful romantic relationships in your life, they certainly are reliable and won't let you down. Carbohydrates include both singular sugar molecules and long chains of them. The simplest carbohydrates, or singular sugar molecules, are called monosaccharides. They are the monomers of which sugar polymers, or polysaccharides, are constructed. There are several different types of monosaccharides, but the most common monosaccharide is glucose, which is a really important sugar in living things. Now, let's look at the molecular structures of these monosaccharides. You can see here that all of these are based off of carbon chains, with some hydroxyl and carbonyl groups attached. We can break monosaccharides down into two groups, aldoses and ketoses. These groups are differentiated by the positioning of the carbonyl group in the monosaccharide. In aldoses, the carbonyl is at the end of the monosaccharide, while in ketoses, they're in the middle of the chain. Although it's definitely convenient to draw them linearly, monosaccharides generally form rings, and this is what alpha glucose looks like in ring form. Before we proceed, I'd like you to note that there are two different types of glucose, alpha and beta glucose. For now, we're going to look at alpha glucose. Each corner of the ring represents a carbon atom. Notice these hydroxyl groups pointing downwards on the two sides of this glucose. This is how alpha glucose binds to each other. These hydroxyl groups on two different molecules come together, releasing a water molecule and leaving behind an oxygen binding the two glucoses. This is called a dehydration synthesis because you lose a water molecule linking them together. You can keep on extending this polysaccharide through this process of dehydration synthesis. Now, let's take a look at beta glucose. Spot the difference between beta and alpha. In beta glucose, this hydroxyl group on this carbon points up instead of pointing down like it does in alpha glucose. Now, that's an issue, because since these hydroxyls are no longer oriented in the same direction, you can't link beta glucose together in a straight line as you can in alpha glucose. So what ends up happening is beta glucose has to link up with every other monomer being flipped upside down, so that the hydroxyls line up and dehydration synthesis can occur. Now, how is this actually relevant to living things? Both plants and animals use carbs as their primary energy source, but they differ in how they store it. Plants store their carbohydrates as starch, which is a polymer of alpha glucose, as granules within their cells. They can come back and get it later by hydrolyzing the starch, or adding water to it, which breaks the starch back down into glucose, reversing dehydration synthesis. On the other hand, animals store their carbohydrates as glycogen, which is a polysaccharide that branches out at multiple points. When demand for glucose increases, say the animal is doing some physical activity, glycogen is hydrolyzed back into glucose for use in the cells. So, how do we use carbohydrates in the real world? First of all, obviously, it's a part of your food. Table sugar is called sucrose, which is synthesized when a glucose and a fructose links up. By the way, fructose is the sugar that you usually would find in honey or in fruit. Another interesting application of carbs is in surgery. Chitin is a polysaccharide that is composed of a modified form of glucose. In nature, arthropods use chitin for their exoskeletons, but due to its pliability and toughness, it is also used as material for surgical thread. Since it's organic, it dissolves on its own, sparing patients the process of removing stitches. Today, we covered the basic molecular structure of carbohydrates, talked about how monosaccharides bind into polysaccharides, and looked at applications in living things and in the real world. See you next time!